Thank you, Russ and Bonnie. Thanks very much for the invitation and giving me the opportunity to be here today and to present this talk. Only 20 minutes for this is very short, but so I decided to put plenty of images then instead of talking a lot. So in human fascioliasis, we are dealing with two species involved in human infection, hepatic and gigantica, two host life cycle, very important, low specificity at the level of definitive host, so with many ruminants and many different groups, equines, pigs, and so on involved, and more specificity at the level of intermediate snail host or vector, as you want, so belonging to the family of the limnates. And within limnates, we have to distinguish on one side the Galbaphosaria group, which includes species transmitting hepatica. On the other side, the Radix group, including species responsible for the transmission of gigantica. And there are other secondary groups which participate very sporadically, except Pseudosoxina columella, which is a species apparently able to transmit both fasciolates. We are talking about a highly pathogenic disease with a high morbidity and even mortality rate if not treated. And that's why WHO Geneva decided to include this disease inside the foodborne dermatodiasis group in the neglected tropical diseases list. Well, this is the only entity which shows a global distribution, as you see here. And here you see the last map we built up in Geneva, and these are the countries presenting reports of human infection. But the problem is not that, is the distribution of the human endemic areas. And here you see the different foci or regions presented in the human endemic area. So when we started with this, the idea immediately was, let's see the common factors, which may explain us why a given area is ready for a human endemic situation, whereas in others you only have isolated or sporadic cases. So we were working about 20 years on that in the different endemic areas of the different continents. I must recognize that the results were really disappointing. So we were unable to find a common factor. And the conclusion was that we have human endemic situations in highly heterogeneous situations here, from very high altitude, from altitudes lower than the sea, unbelievable, and also from the physiographic point of view, different continents in islands, and unbelievable. So the conclusion was the opposite. The conclusion was that we are in front of the vector-borne disease presenting the widest latitudinal, longitudinal, and longitudinal distribution known. So at least three conclusions we got is, first is gigantica is only present in Asia and in Africa. It's absent in the Americas and the Oceania because of the absence of limnates of the Radix group. Second, many water bodies do not allow for the transmission of the disease because they are saltic, such as the Caspian Sea or the Lake Titicaca. And the last conclusion, very important, there is absolutely no correlation with animal fascioliasis. So you can have very high human prevalences in areas where at veterinary level, the problem is neglected. So now, well, we are facing three main questions. First is the surprising geographical distribution of these human endemic areas. The second, the surprising conservation of the nuclear and mitochondrial DNA markers. And very recently, the surprisingly large heterogeneity at the level of the whole genome of Fasciola hepatica. Well, let's see if we are answered to. First, a little bit paleontology. Well, we did plenty of studies during many, many years, and the conclusion was the paleobiogeographic origin of Gigantica should be looked for in the lowlands of East Africa, most probably thanks to the transmission of Radix natalensis, which is a very old species, and in the warmest, very warm climates in these lowlands, and using very old bovines of different groups in the Miocene, so around 20 million years ago. Hepatica completely different, the origin is completely separate, and all suggests that it appeared in the Near East, probably in altitude, in a cooler area, in different ruminants, so mid-sized ruminants, the ovicaprines, so sheep, goats, and so on, and adaptation to Galba truncatula, which is a snail more adapted to cooler climates. And this happens thanks to the migrations of the ruminants which took place in the Miocene from Africa, and the fauna entered in that way in the Near East. So this is the pre-domestication period. Now we enter in the post-domestication period. No? And this is concerning Gigantica, our conclusions about the spreading ways followed by these species. No? So here is the origin and how it spread around. So first, very important, the old movements and commercial activities related to the spread of these species in Africa. Unfortunately, the knowledge about all that is not so much in detail as we have from other continents, but the results really fit with the DNA and genetic and archaeological studies we performed. Very, very important is the crossroad we have in the Near East, in the so-called Fertile Crescent, in the old Mesopotamia and so on, where the domestication of livestock started, no? Around 10,000 to 12,000 years before Christ. No? And very important, at the end of that period, there was an overlap with the first dynasties of the Old Egypt, and there was a big exchange of livestock between both civilizations. This is new, not yet published. This is our recent results, the finding of eggs in human coprolites and archaeological sites in the Iranian Zagros Mountains. It's very important, we have found eggs of the two species there, so again, all fits. Also very important is the later dynasties of Egypt had a great exchange of merchandises and so on with Nubia, a little bit southward. And we have found that there are findings of eggs in human coprolites in that archaeological sites too. Afterwards, we have been obliged to play a little bit in the Indiana Jones. So here you have the Egyptian Museum of Cairo and the gold room of Tutankhamun, and we were able unexpectedly to find this bracelet constituted by 24 gold pieces. And when you look at these pieces, they are really Radix natalensis. They didn't know at the museum they had this. There was no label on that. And they are so perfect that the snail is surely inside, and they are gold covered. Going on with our Indiana Jones performance, we were looking for information in different antiquaries and old libraries and so on, and again, success. We were able to find the very old booklet of Los, which included the description of Fastiola hepatica aegyptica, and this is 2 centuries ago, and which tells us that Gigantica was already there in that period. So all fits. Now, Asia. The Silk Road played an unbelievable roll there. The seal roll started 138 years before Christus and was working during 15 centuries. Can you imagine 15 centuries, cows, zebus, camels, dromedaries, transporting fasciola and the limnates attached to the legs from east to the west. And very important here is that when you look at the ways of the Silk Road, there are given roads northward from the Himalaya and others which are southward from the Himalaya. And this is very important from the point of view of temperature. So also suggests that Gigantica was more successful in spreading through the southern roads and Hepatica evidently through the northern roads. Of course, afterwards, with ships, the Gigantica was able to spread to Madagascar, to the Philippines, and to Hawaii, and we, in many of these areas, we had human endemic areas. Now concerning Hepatica in the post-domestication period, there is a first step of spread in the Old World, and you see here through eastern part of Africa. We are now analyzing, in collaboration with RAS, the protopoison spread in Tanzania and South Africa. And the results, for the preliminary results fully support, again, the ways. And here you see it has been a spread in the Mediterranean and Europe, but also into Asia. Well, when we look at the archaeological findings, it's very interesting to see that fasciola Hepatica eggs were found in many coprolites in different archaeological sites from the Mesolithic and Neolithic, so around 5,000 years before Christ, no? Also so the Bronze Age and so on. And there are different countries of European countries in which such findings have been made. And very important, never found eggs in human coprolites or in mammals in the New World. So first the Mediterranean, the Phoenicians undoubtedly play a very, very important role. The Phoenicians were always carrying with their sheep and goats, so mid-sized ruminants. Afterwards, the three great empires, says the Greeks, perhaps they did something, but they were not giving livestock so much importance, opposite to the Romans. The Romans gave a lot of importance to agriculture and to livestock, mostly in the eastern part of Europe. It is today in the eastern part where we have more problems with human fascioliasis. And afterwards, also the Arabs, the Arabians, Muslims were 800 centuries in the Iberian Peninsula, and their management of sheep and horses is well known. Also, the leather roads surely played a role in the spread of Hepatica more northward. In Asia, again, for Hepatica, again, the Silk Road, and as I was saying before, here more with the participation of camels. Camels were mostly used in the northern ways, whereas dromedaries were more used in the southern ways. The spread of Hepatica, this is very interesting. In the continent of Africa is Fasciola gigantica, but we have Hepatica in all this eastern part of the continent. And when you look at the history, you see the relationship of Old Egypt, Nubia, afterwards the Kushites, afterwards the Protocoisans, until arriving up to South Africa. And we have many human cases and even fatal human cases today in South Africa. So afterwards, the problem that we are facing today with hybrids. So here we have to differentiate between the pre-domestic period, which is from 19 million, 20 million years, all this very long period in which both species were geographically separated, and the last 12,000, 10,000 years in which, thanks to human activities, the two species were obliged to overlap and therefore the possibility to start with hybridization. Afterwards, yeah, very important is the colonization of the New World with ship by the Spanish and also Europeans of Oceania, because in the ship they were transporting always livestock with them. And this was the way in which Fasciola hepatica colonized the New World. And very important, the discovery of a Limnaea syracensis, which has proved to be an excellent livestock movement marker and which also shows to follow the same spreading routes. And our last contribution, very recent, we have been able to show how Hepatica was able to colonize the eastern lowlands of South America from the highlands of the Andean Conference, this last contribution. So the three questions, here are the answers of the three questions. Surprise, regarding the geographic and distribution, this is linked to the mankind's history, not to other questions. Second, the conservation of the DNA marker is due to a worldwide diffusion only in recent post-domestication times. And regarding the surprising large heterogeneity of the genome, we are dealing with organisms with genomes perverted by accelerated evolution to human influences. That's all. Thank you very much.